So we are looking at the phases of mitosis, the illustration of each, and the major events. So you have the first thing you should be looking at is this part of your notes. So as I'm going, you can draw the pictures first, and then we'll add in the major events up here. So we'll be doing these in order. Okay, so mitosis is made of basically 5 phases. Sometimes you see it as 4 and cytokinesis off by itself. But prophase, metaphase, anaphase, telophase, and then cytokinesis. So the best way to remember the order is you can just say PMAT. You can say please make another taco. Those are the 2 ones that I know. Okay. So this is prophase. We'll be doing these in order. Over here, this is prophase. Right here, in a normal phase, we're in interphase. So this is when the cell is not dividing. And notice the difference between, this is the nucleus right in here. Notice the difference between the nucleus here and as it is here. So the first thing that happens during prophase is chromosomes condense. So here in interphase, the chromosomes are all spindly, all just spaghetti in there. And then they condense into the X shape during prophase. And then you can see them much more easily under a microscope. So chromosomes condense in prophase. The other important thing is these, the centrioles that you learned a long time ago. Remember, they look like churros. They start moving. So they're normally just wherever in the cell. The centrioles start moving to opposite ends of the cell. You see it here. So the centrioles move to opposite sides of cell. And if you remember, the definition of centriole from the cells chapter is aiding in cell division. This is now we're finally getting to learn what they do. They send out these lines that you see here called spindle fibers. And the spindle fibers attach to the chromosomes and move them around. So the centrioles move to the opposite sides of the cell and start sending out spindle fibers. So some places, instead of saying spindle fiber, they say spindle apparatus. Some just say spindle. As long as you see the word spindle, it's the same thing. Okay, so the chromosomes condense, the centrioles start moving and sending out the spindle. The other really important thing that happens during prophase is the nuclear membrane goes away. So right here you have a very solid nuclear membrane holding the nucleus together. You have it here too, but as you see here, the nuclear membrane starts breaking down. So that's important because the DNA then spreads out all over the cell and it's able to split the whole cell and not just the nucleus. So either dissolves, goes away, whatever. Okay. So these are the 3 main things in prophase. Okay. Then we have metaphase. Okay, so during prophase, the spindle fibers were sent out and they connected to the chromosomes. During metaphase, those spindle fibers pull on the chromosomes and start moving them around until they reach the middle of the cell like this. So the big major event for metaphase is spindle fibers pull the chromosomes to line up in the center of cell. In the center, or you see midline a lot, or equator even sometimes. Okay, so meta, like middle, line up in the middle. And that's all that metaphase is. It's metaphase is like a split second where they're all lined up in the middle. Okay. The next one is anaphase, and what's going to happen here is the chromosomes break in half and the spindle fibers shorten like someone reeling in a fishing line, and it splits the chromosomes and they start moving away like this. Okay, so this is anaphase. And you can A in anaphase, like away, they're moving away. So what's happening in anaphase is sister chromatids are split. And they move to opposite sides of the cell. And that's really about it. This is the end of anaphase right here. They're all, see how the centriole kind of reeled them in like a fishing line? The spindle fiber gets shorter and shorter. These lines in here are just showing the movement. And then eventually you have all the chromosomes here. And then the last thing to happen is telophase. So all that happens there ends up looking like this. All that happens in telophase is the chromosomes uncondense back into spindly stuff. The nuclear membrane comes back, so it's preparing to be a regular cell again. So nuclear membrane returns. Chromosomes decondense or uncondense. And that's telophase. So now you have two little regular nuclei. They're back to normal. There's a membrane. The DNA is not condensed. The only thing that's left to do is split the cytoplasm. So remember, we started as one cell, and there's all these organelles all over the cell. So the organelles have to be divided too. And the last thing, some people call telophase the end of mitosis because we're back to a normal nucleus. But the last step that has to happen is this has to break. Okay. So this is cytokinesis. Okay. And that, now you have two new cells, so it broke. So splitting of the cytoplasm. So look, cytokinesis splits the cytoplasm. Kinesis means movement, like a kinetic energy or something. So this is the cell is moving apart. So now you have the whole cell separate. Forgot to tell you one thing about telophase. Telophase sets up this kind of like perforation, and that's called the cleavage furrow. This right here, how it's pinching in. So it's giving cytokinesis the place for it to break. That happens during telophase as well. Okay. So you should be finished with this little chart right here. Should have been pretty easy to follow along with me. Now the rest of the questions I am going to have you do by yourself. This one should be really clear. These are basically opposites. And this question is asking you about cytokinesis in animal cells versus plant cells. Okay, and right here, just explain why. I'll tell you what the difference is tomorrow, but you need to be understanding why there is a need for that difference. And this I'll do with you just in case you haven't done it already. So a chromosome is a bunch of DNA. DNA is swirly like this. Okay, so this whole thing this whole thing is a chromosome. Each half going this way is a chromatid. So that's one chromatid, and in the blue is one chromatid. Okay, they're called sister chromatids because they are actually identical to one another. That's important for the reason why mitosis works, because if this half goes off to the right and this half goes off to the left, they're going to be sending identical copies of DNA to the new daughter cells. Why is everything sister, daughter? I don't know. All right. Then holding the two sister chromatids together in the middle, like a little button or a snap, is the centromere. Centromere. So you have to be careful. You have a centriole and you have a centromere. The centromere is the button. All right. Okay, so the rest of the questions, you need to do these. Okay. You can do this by yourself. Think through them. It's all about chromosome numbers. Okay. Wonderful.